Transport and Biological Systems, Lab 8. Recall the equation for cellular respiration. On the side of the reactants, you have glucose, oxygen, and on the side of the products, you have carbon dioxide, water, and the high energy molecule ATP, energy and heat released. Now, on the reactant side, for cellular respiration to take place, you need glucose, and glucose is going to be provided by the digestive system, as we saw in Lab 5. Oxygen is also needed, and the respiratory system will take care of delivering oxygen and making it available for our cells to use. Both glucose and oxygen are coming from outside the cell into the cell. Carbon dioxide on the product side is a waste product, and it needs to go from inside the cell to the outside of the cell. These respiratory gases need to move according to diffusion. So gas exchange involves ventilation, diffusion, circulation, and respiration. Ventilation is the exchange of gases. Oxygen is inhaled, carbon dioxide is exhaled. So the respiratory system takes place of ventilation. Oxygen that is inhaled will cross the respiratory surface, the respiratory membrane, and be picked up by our red blood cells in our circulatory system. Carbon dioxide, which is a waste product, is going to be exhaled out. The oxygen is delivered to the tissues at the tissue level, so via diffusion. So it could be utilized in cellular respiration for ATP production by the mitochondria. So oxygen has to go through the cell membrane and diffused in. Carbon dioxide diffuses out, transported via the circulatory system, and exhaled as a waste product. At the tissue level, we not only have oxygen diffusing out of the circulatory system. This is a capillary, the smallest of blood vessels, but we also have nutrients such as glucose that's utilized in cellular respiration. Both oxygen and nutrients are going to diffuse into the tissue cells for utilization, such as cellular respiration. And waste products such as carbon dioxide and other metabolic waste products have to diffuse out of the cell, picked up by the cardiovascular system, transported to the appropriate organs for excretion. Diffusion is the movement of matter down their concentration gradient from a region of high concentration to a region of low concentration until equilibrium is reached, which means that the solid molecules are evenly dispersed and they're moving equally in all directions. The driving force of diffusion is kinetic energy. Kinetic energy is the random movement of molecules. A physiological example of diffusion is that gas exchange, that movement of oxygen and carbon dioxide between lungs and the blood, and between blood and the tissue cells. So here is a close-up example of what's happening in the alveoli. Those are the air sacs of the lungs, and a capillary, the smallest of the blood vessels. Oxygen moves from high concentration, it was just inhaled, down its concentration gradient, picked up by red blood cells in the blood capillary, and then carbon dioxide is in high concentrations in the capillary, so it will diffuse down its concentration gradient into the alveoli, the air sac, where it will be exhaled. Similarly, you'll see the example of diffusion in the dye experiment, where the dye molecules will diffuse from areas of high concentration to areas of low concentration until they evenly disperse, and then they'll be moving in all directions. Now, diffusion is passive. It does not require energy in the form of ATP. There are several factors, though, that affect diffusion rate, how fast over a given period of time diffusion takes place. The distance will affect diffusion. Shorter distances equals faster diffusion. In your respiratory membrane, the gases are going across a membrane that's less than a micron, half a micron in distance. Molecule or ion size affects diffusion. Those gases are very small. Smaller the size equals a faster diffusion. Temperatures affect diffusion. Higher temperatures equals faster diffusion, which you will see in the dye experiment. Concentration gradient, steeper gradient equals faster diffusion. And lastly, electrical forces, attraction of opposite charges and repulsion of like charges. Diffusion of substance in and out of cells saves the cell a great deal of energy in the form of ATP. Because there's energy, potential energy, in that high concentration of molecules on one side of a membrane in comparison to the other side of the membrane. So the molecules are moving down their concentration gradient. This potential energy then becomes kinetic energy, energy of motion, going down the concentration gradient. It saves the cell a lot of ATP energy. This is a passive transport. That movement of the molecules down the concentration gradient, which describes how oxygen and carbon dioxide are moving across a membrane and how glucose moves down a membrane and other molecules. Here, glucose actually needs the assistance, so to speak, of proteins embedded in a cell membrane. So this is a special type of diffusion called facilitated diffusion. Facilitated means it's being helped, and the assistant are proteins embedded in the cell membrane. The permeability of membranes. Materials may cross the plasma membrane through active or passive processes. Diffusion, again, is a passive transport system. It does not require energy in the form of ATP. And then facilitated diffusion, that's going to be called carrier-mediated transport, where a protein embedded in the cell membrane will facilitate the transport of molecules. And those are also going down the concentration gradient from high to low. Active transport refers to the fact that the cell would need energy in the form of ATP for molecules to move across the cell membrane, and that ATP will drive molecules across a membrane regardless of their concentration gradient. In fact, it would transport molecules against their concentration gradient from low concentration to areas of high concentration. This also involves carrier-mediated transport, so proteins that facilitate in transport, but they need the ATP energy for transport of substances, and vesicular transport. Vesicular are for large proteins or amino acids. That involves molecules inside a vesicle that are going to exit the cell via exocytosis, and that requires energy in the form of ATP as well. Osmosis is the diffusion of water through a semi-permeable membrane. It is a passive transport system. It does not require energy in the form of ATP. If we look at the first diagram, we have a U-shaped tube, and in the middle of the tube, we have a membrane representing a selectively permeable membrane, such as our cell's membrane. That selectively permeable membrane means that there are certain molecules that could cross the membrane and certain molecules that can't. Water is a small molecule and is able to cross the membrane, but other molecules, bigger molecules, are not able to cross a membrane, so we'll call these non-penetrating solutes. So on the left-hand side of the tube, we have less solute in comparison to the right-hand side, we have more solutes. If we have less solutes, it means we have more water molecules. If we have more solutes, it means we have less water molecules. Water will move from high concentration of water molecules to low concentration of water molecules. Another way to view this is that water will move to dilute concentrated solutions. Again, solution are solutes dissolved in a solvent. In this case, the solvent is water. So water is moving to dilute concentrated solutions. And you will see that on the right-hand side of the tube. Water levels rose on the right-hand side, and this was due to the higher number of solutes in the right-hand side. For one of your experiments, you're going to view the effect that solutions have on cells. You'll look at one video that shows plant cells, and you'll look at what happens to red blood cells when placed in solutions that are hypotonic, hypertonic, and isotonic. Let's explore the term tonicity. Tonicity terms hypertonic, isotonic, and hypotonic. Tonicity terms are relative. They are used when comparing two solutions that differ in concentration of non-penetrating solutes. These solutes that cannot penetrate a semi-permeable membrane. So here we have two containers represented by these triangles, and in between them they have a semi-permeable membrane. On the left-hand side, we see more solutes represented by the white dots, and on the right-hand side, we don't see any solutes. The apparent space is filled with water, both in the right-hand side, and also the solutes are dissolved in water. So on the left-hand side, we have lots of solutes, so this is hypertonic in comparison to the right-hand side, that's hypotonic. Again, we could reverse the way we view this and just say that the right hand side is hypotonic in comparison to the left hand side that's hypertonic. Again, if there's this space is representing water, water will move from an area of high water concentration, low solute concentration, to an area of higher solute concentration and low water concentration. Or you could view it as water's moving to dilute concentrated solutions. Isotonic solutions have the same concentration of non-penetrating solutes. And so water is equally going to be moving back and forth. For the lab objectives for Lab 8, you will have the opportunity to explore the transport mechanisms just discussed, like diffusion and osmosis, and the effect they have on cells and the cellular response accordingly. You're going to look and apply these principles to physiological functions, and to remember that in order for diffusion to take place, we need these molecules, oxygen, glucose, ions, amino acids, etc., to be transported in bulk. So these molecules need to be transported in bulk by the cardiovascular system. The cardiovascular system is made up of the blood vessels, the blood, and the heart. And so we're going to explore the anatomy and the function of the heart. The heart is a dual pump system that's going to pump oxygenated blood everywhere in the body. You're going to identify the parts of the heart and describe their function. And accordingly, you will apply concepts learned to making healthy lifestyle decisions. Activities for Lab 8, pre-activity, diffusion. You'll watch a video on diffusion and the factors that affect diffusion rate. Activity 1 will show you observing cellular response when placed in various environments. Activity 2, naked egg experiment. Activity 3, tracking your pulse. And Activity 4, understanding the heart. Activity 1, you're looking at what happens to plant cells when they're placed in varying concentrations of solutions. So you're going to make a prediction before you watch the video. What would happen to a cell under 0% salt solution? 0% solution would be cells in deionized water. Deionized water, absolutely no solutes. What would happen to these plant cells when placed in 1% salt solution? What would happen to the plant cells in 10% salt solution? Draw your prediction. Then watch the video, osmosis and the red onion cell. And after watching the video, during watching the video, record your results in your Lab 8 worksheet. Activity 1, animal cell, observing response on a cellular level for animal cells. You're going to watch the video and record what happens to red blood cells when placed in 0.9% salt solution, 10% salt solution, and 0% salt solution. Describe the structure and describe what happened to the cell. Also draw what you see in the video for cells that are in 1%, 10%, and 0% salt solution. Draw a sample. You'll see many cells in that video under the microscope. This represents here on the lower left-hand side a typical red blood cell. Red blood cells are about 8 microns in diameter. They are disc shape with the center caved in. This allows for greater surface area. Remember that one of the factors that affect diffusion rates is an increase in surface area. You increase surface area, you increase diffusion. And so the shape, the structure of the red blood cell lends to its function. When placed in various solutions, you will explore what happens to the integrity of that function. And a structure that's altered will also have altered function. Activity 2 is called the naked egg experiment. So you're going to observe osmosis in an egg, that movement of water. So the materials that you need are 4 raw eggs, 4 mason jars or glass clear containers with a lid, white vinegar enough to cover all 4 eggs in a jar, 100 grams of salt, 4 liters of water, and paper towels. If you do not have the materials at home, do not worry. You do not have to perform the experiment at home. You will watch the following videos of the experiment performed and you'll answer the questions according to the videos that you watch. But it is fun if you actually learn by doing, and so I highly encourage you to try the naked egg experiment. Activity 3 is tracking your pulse. Remember that diffusion matters, but it cannot take place unless you have the circulatory system functioning and delivering that oxygen that was picked up from the respiratory system, that glucose that was picked up from the digestive system, and delivering it to all cells. And so the heart becomes very important in ensuring that molecules are being delivered to the right destination. Now, heart rate is how many times is your heart beating in 1 minute, because each beat represents a movement of blood delivering that oxygen, that glucose to your cells for cellular respiration to take place, so ATP could be produced. So to assess and to determine your heart rate, sit quietly for 3 minutes. Measure your pulse by pressing on the radial or carotid artery. You will use your middle finger and your index finger. Your radial artery, the radius is the bone located on alongside the same side of the thumb. So you will feel your radial artery on your wrist along the thumb, and then wait quietly to feel for that pulse, or you could look up the carotid artery, which is on either side of your trachea. Once you feel your pulse, you'll wait and time yourself 30 seconds. Count how many pulses you feel in 30 seconds. When 30 seconds is up, multiply that number by 2, and that will give you how many times your heart is beating per minute. Once you determine your resting heart rate, you will determine your heart rate after exercise. So you're going to conduct some sort of activity. You could dance, you could run, you could do jumping jacks. You decide, for about 5 minutes. Then you're going to take your pulse and record it on the 0-minute column. So immediately after you exercise, record your heart rate at 0 minutes. 2 minutes after your exercise, record your heart rate. 4 minutes after your exercise, record your heart rate, etc. 6 minutes, record your heart rate. The faster your heart returns to resting heart rate, the better shape you're in. So that's one determinant of, where your physical level is that you could always see how soon your heart could go back to that resting heart rate. Now, if you have any heart condition or health condition, please refrain from participating in this activity. Activity 4 is understanding the heart. Again, the heart is a dual pump that's allowing the transport of oxygen and glucose and nutrients and movement of waste products so they could be excreted. So the heart is a dual pump. It simultaneously pumps blood to the pulmonary circuit, to the lungs, so it could become oxygenated, and to a systemic circuit everywhere in the body, so it could deliver oxygen and nutrients everywhere in the body. So for this activity, you'll watch a video that shows you the anatomy of the heart, the external anatomy of the heart, the internal anatomy of the heart, and blood flow through the heart back to the heart and to the whole body, the systemic circuit. Importance of Lab 8. We just saw that diffusion matters because we need that oxygen delivered. We need that carbon dioxide picked up and exhaled. The osmosis, our body, we are mostly water, and our cells are water. So our cells have solutes. So that our cells inside is the solution. Outside there's the solution. Water is the solvent. Water is constantly moving in and out of cells. And so we want to see the effect of solutions on water movement. Because remember, according to that red blood cell video, what happens if too much water goes into the cell or too much water exits the cell? The cardiovascular system, again, very important because it's transporting. It's our transport system. Oxygen is needed by the tissues. The cardiovascular system allows for bulk transport, oxygen being delivered to the tissue level, and carbon dioxide delivered to the respiratory system for it to be exhaled. Glucose, nutrients, vitamins, minerals being picked up from the digestive system, delivered to the cells, the tissues that need them. Now, atherosclerosis is the formation of lipid deposits in blood vessels, and it will damage blood vessels. Blood vessels are part of the circulatory system, and they allow for the transport highways of the circulatory system. So if there's a buildup of plaque, fat deposits in the blood vessel, it's going to narrow the diameter of the blood vessel, meaning that it's going to decrease the amount of nutrients in our blood that's delivering that vital oxygen and glucose for cellular respiration. The respiratory system, it's assisting in ventilation. That's oxygen in, gas exchange at the level of the respiratory membrane. Remember, diffusion is dependent on surface area. If you're a smoker, you're decreasing the amount of surface area because the alveoli are damaged and they break. Small little air sacs become large air sacs. You're decreasing surface area, and surface area affects diffusion rate. So if you decrease surface area, you're going to decrease diffusion rates. And that means you're going to have less oxygen being transported in and a harder time getting carbon dioxide out and exhaled. So cardiovascular disease is the number one cause of deaths in human. It could lead to heart attacks, myocardial infarctions. Risk factors for cardiovascular disease include age, tobacco use, poor diet, high-fat diet, high blood cholesterol levels, obesity, lack of physical activity, and your genes. So apply what you learn, apply it to your life, and I hope you enjoyed the lab. What are your next steps to complete the lab? Read the instructions for this lab. Read the instruction PDF for this lab alongside Lab 8 worksheet. Schedule time when you will work on the lab. Start on Monday when they become available and schedule the time that you will work on the lab. Use the tools given to you in your Blackboard to complete the lab, including this video. And lastly, attend your Zoom meeting prepared with questions regarding this lab, which means congratulations, you are now ready to start Lab 8. Begin the lab before your Zoom meeting.